Good afternoon, one and all respected over here. I, Dr. Asha Patel, Associate Professor, Department of Pharmaceutics, Parul Institute of Pharmacy, Parul University, herewith welcome you all on a virtual mode at webinar platform of Parul University. Today we are going to have one of the person from uh, MSN Laboratories, Hyderabad. Herewith I would like to welcome you, sir, for today's session on fundamental aspects of ophthalmic drug product development, fundamental aspects to regulatory consideration. With a short, brief introduction of today's speaker, Mr. Nithun Shaha did his master degree in pharmacy from Dr. MGR Medical University in 2005. He has more than 16 years of experience in pharma R&D. He has worked for different companies like Sanofi Aventis, Alchem Laboratories, Virgo Pharma Research, St. Louis Pharma, etc., as well as Amneal Pharmaceuticals, Ahmedabad. He is having experience in the formulation development and technology transfer of solid oral, injectable, inhalational, and ophthalmic dosage form. He has worked for several dosage form for several regulatory markets like US, European market, Canada, Russia, etc. Presently, he is working as an Assistant General Manager, Formulation Development, Department of Complex Injectable and Ophthalmic Product at MSN Laboratories, Hyderabad. Sir, with this introduction, I would like to welcome you over here at Parle University webinar platform. Over to you, sir.
Thank you. Good afternoon, all. Myself, Nithun Shaha. I think already ma'am given that introduction from me. And today our topic is fundamental aspects of ophthalmic drug product development. Just a minute, wait. For the process, for drug development process, for all the drug product development, like injectable, any sterile products, when you develop for inhalation, ophthalmic suspension, emulsion, injectable suspension, emulsion, there are some steps we have to follow. That first, discovery concept for the development, then preclinical research, and step 3 is the clinical research. Then we have to develop our product for FDA, and for US that is the FDA. Then if you go for that Europe, you will go for that MHRA and EMA, and as well as for Russia, yes, there is other regulatory agencies. After that, FDA review, they will provide their comments for our development. What are the lack and what are the data they have reviewed. If there is something is missing, they ask for the data. They have to submit according to their review and our submission. They will approve our product, and followed by, if the patent will be expire, then we can easily go for the market. For step 1, that discovery concept and development for research for a new drug begins in the laboratory. First, for our company, some API they will give the API. After for receiving the API, that our business development team, they are looking for that what are the doses from we can make from that API, that injectable, ophthalmic, all those things, and what are the market potential in different US, Europe, like that. According to that, they will issue one PIF, product initiation form. According to, will act. After that, whatever will go for the literature search from different sites. Basically, actually, we will go for FDA site. For FDA, first we have to follow that orange book, whether the drug product, what is the RLD. RLD means the reference listed drug, and followed by that any RS is there, reference standard is there or not. Then what is the approval date of this product? What are the expiry? Is there a patent is there and patent expire or not? After that, we'll process. We'll go for the literature search. Literature search means that there's some documents, whatever RLD already submitted to the FDA. Just the document is summary basis of approval. Summary basis of approval. In summary basis of approval, certain contents are there. Therefore, that chemistry review of the product, that product labeling, pharmacological review, clinical review, then stability review. All these document we can access from that site. After reviewing all these things, then we will go for the preclinical research, drug undergo laboratory, animal testing, and from animal testing, some answer a basic question. Some basic questions from we receive from the third party. Then whether our product is sterile or not, whether our license is there. Then we can proceed the batches, all these things. Then we go for that animal toxicity. If animal toxicity is okay, as well as our development is ongoing side by side, and we go for that product different stages. We will come later. After that, if animal toxicity is okay, if there is any requirement for bio, then we go for bio, or if bio is not required for suspension emulsion, then we can go for the Q3. Q3 means the in vitro characterization of the product. Then people make sure that they are safe and effective. And if the already that product is approved in the FDA, then safety efficacy already established. If there is any change of excipients, then you have to establish that whether your product is safe or effective. If there is a change in preservative buffer, and another one thing that one literature is there, 21 CFR, there they have already told if you change any buffer and preservative, all these things, you have to prove your stability with respect to RLD. You meet with specification with respect to ICH guidelines as well as RLD comparison, then you can proceed for the filing. Then for FDA review, FDA, when you submit the data after our exhibit batches are when completed, then they will review. After review, they will give the comments already told. Then finally, we have to go for the market. Formulation development for ophthalmic products. That formulation in ophthalmics, when you go for the ophthalmic products, there is some active pharmaceutical ingredients and other inactive ingredients. In other inactive ingredients, if you go for the liquid formulation, there's some buffers, then pH adjusting agent. And if you go for the suspension, then there is viscosity modifier agent. And obvious, there is a buffer as well as pH adjusting agent. Apart from that, viscosity modifier agents of some HPMC, HEC, xanthan gum, we can use that for the development. That each and every inactive ingredient in the formulation is to serve the specific purpose to ensure the product performance and conformance. Then each and every inactive ingredients, when you told that the formulation is used to serve specific purpose to ensure the product, then when we go for the FDA for submission, then we have to mention that xanthan gum used for the viscosity modifier. If diabetic sodium phosphate, this is the buffer. You have to mention. Then you will accept. Otherwise, you will comment whether the material is used for or not, what purpose we have used. An ophthalmic formulation could be solution, suspension, ointment, and emulsion. And typical eye care product is sterile, nearly isotonic, has some buffering capacity, contains antimicrobial agents, and packaged into a suitable tamper-evident multi-dose dispensing system. Here we have mentioned the multi-dose dispensing system. Multi-dose dispensing system means that particular product you can use up to 28 days, 32 days, as per your in-use stability of the product. In-use stability of the product. Otherwise, you can go for that unit dose of product. Unit dose means you have that respule, that's different pack volume with 0.25 ml, 0.5 ml, 0.3 ml according to the dose. You have to use that and throw it. So for unit dose product, there is some challenges because so many companies don't have that machine for particular unit dose products, and this is very costly. If you go for multi-dose product, there is some preservative. There is several pack size, 5 ml, 10 ml, 15 ml. There is no requirement because 3-piece line. If you see, every company who is having the ophthalmic product, they are having a 3-piece line always. After, when we decide that we have to develop the ophthalmic formulation, then we have to some regulatory norms are there. For injection, if you see product label through the FDA, the mention of excipients, the quantitative and qualitative mention of excipients in each and every injectable product. If you go through the level of ophthalmic product, there has a mention of qualitative composition. There is no mention in quantitative composition. Then how you can decide that your product is qualitative and quantitative similar with the reference listed drug product. So for that, when you initiate for ophthalmic product development, first you have to do the reverse engineering of innovator product. Whatever excipients they have used, how you can quantify of them that excipients through different methods, that HPLC, UPLC, UV, all these things. According to that, we have to set 3 formula. That formula will be plus minus 5% with respect to RLD formulation. Then we have to submit the 3 formulation to FDA for approval, whether our formulation is plus minus 5% with RLD or not. If yes, then FDA told us that go ahead, you can do the formulation. If no, they reject our control correspondence. Then again, we have to think, we have to search the literature search, then go for the reverse engineering again. We have to quantify again. Then for quantification, what we have to do? First, we are using some from the literature, some quantity we have mentioned, known quantity we have used in our formulation. Then give to our analytical department. For analytical department, do the analysis with respect to the standard. If we give that 5 mg per ml sodium chloride in solution, suppose 10 mg per ml xanthan gum, like that, a known quantity we have given to our analytical department. With HPLC method, UPLC method, another analytical method that they are using, and they quantify that our 10 mg with standard value, whether they will get or not. If their method is robust and their method is developed, then they run the RLD again with respect to our method developed method. Then we can know our reverse engineering that quantity, whatever the quantitative composition of RLD. During formulation development, choice of excipients of buffer based upon the physiological comfort and product stability, preferably within a proven track record with FDA. The ideal pH of an ophthalmic formulation 7.4, equivalent to TR fluid. Maximum of the ophthalmic product targeted the pH near about the 7.4. Somewhere some product that having pH less than 5, less than 4. If you go for the ciprofloxacin dexamethasone, if you see the tobramycin dexamethasone, because the solubility of the drugs, drug substance depends on the pH of the solution. If you not maintain the pH and definitely that less than the 5, that API will comes out from the solution, and it will not safe for the patients. So for some ophthalmic products, for the nature of the API and the solubility, we have decided the pH, and already mentioned if we go for that ANDA product, for ANDA we have to, ANDA means abbreviated new drug application, we have to follow the RLD. Then the pH already mentioned in the RLD, you can follow for that. In dosage form applied to the eye, solution, suspension, emulsion, ointments and gels, strips, different injection, insert, and implants. And most of the companies, they are doing the solution. Solution is very much easier. And for suspension, this is too tough. Suspension and emulsion, too tough means there's some instrument is required. When you go for the suspension product, if the particle size is less than 2 micron, and having the bimodal peak in your Malvern particle size analyzer or Beckman particle size analyzer, then you can easily decide that the particle size reduction innovator is done by that milling process, because there is a bimodal peak and the particle size less than 2 or less than 1.8. Very fine, very fine. If the particle size is more than 3, then you can use easily high pressure homogenizer, microfluidizer, high shear homogenizer, like that. And for emulsion is too tough, because for emulsion, when you go for that emulsion for FDA submission, in vitro characterization is the most important for suspension and emulsion. For suspension and emulsion, because when you go for that suspension, you have to maintain different parameters, that G3 morphology, that viscosity at different shear rate, particle size, population bioequivalence, and physicochemical characterization. All these things have to matching with RLD, and population bioequivalence with plus minus 5% you have to pass. So in population bioequivalence, if you go with the guideline from the FDA side, they have mentioned you have to compare the particle size, that's span value as well as D50, or otherwise Z average and PDI. You have to do the statistical analysis and then submit the data whether your product is compared with the RLD or not. Then they will review, and accordingly they will approve if your data is passed. For emulsion, I have worked for that 2 or 3 emulsions, that nano emulsions. This is too tough, because that emulsion is depends not only in machines, that process. Some process is there, that ratio of oil, that water, that pressure, temperature, every parameter is playing an important role for the emulsion preparation. Ointments also too tough. If the ointment is that if the some API is suspending form, that tobramycin, dexamethasone, all these things. Gels, I have worked for the low tetradenol gel. The tough for gel is the viscosity of this gel is 1400 CPS. The huge viscosity. So due to viscosity, during filling, this is too tough, because if some air bubble is generated during filling, there is too difficult to maintain the accurate fill volume and also accurate delivery. So for viscosity, for gels, the important thing is maintain the viscosity and filling parameter during filling in container. The strips totally different to diagnosis some disease in eye, and some injection is there, insert is there, and implant is there. Nowadays, not all the companies, that big companies, they are working with implants and microsphere also. They are working for eye microsphere. They are working from liposome also, for eye liposome. For novel ophthalmic dosage form, that in situ gelling system. For in situ gelling system, that what it will act in ophthalmic, that improve the retention of the formulation in your eyes, and the release the product is slowly, like as a sustained release. For some product, if you go for the ciprofloxacin, dexamethasone not soluble, not easily soluble with ciprofloxacin, then sometimes they have used a cyclodextrin, gamma cyclodextrin, beta cyclodextrin according to innovator PIL or formulation stability, to improve the solubility and increase the viability of the product. Micellis formation, for micellis, improve the absorption and deeper penetration to the eye. That liposome, for liposome, the advantage of this corneal accumulation and sustained release of the product. Nanoparticles improve penetration and improve absorption. And non-aqueous liquid, not that much, but some emulsion, that cyclosporine, that diphenylprednate, all these things, they are the emulsion oil based. So for whether the API is soluble or not in water, they will go for that oil solubility, and after that they will prepare for the emulsion. For formulation development, what does a formulation scientist do? First, the literature review. I have already told, if you develop for US market as well as their regulatory sites, we have to follow their guidelines. If you go for that US market, you have to be that formulation according to the RLD with plus minus 5% of the concentration. The same thing also happened for Europe, and for Canada market, the same thing. You have to establish that your product is comparable with RLD, and physiological compatibility is there, that pH, viscosity, and buffer capacity, specific gravity, you have to maintain according to the RLD. So for that, first, if you go for the US, that site is FDA CDER, Central Drug Evaluation of Research. If you go for that, first you have to go the Orange Book. Whatever product you want to develop, that your product, who is the RLD, which company is the RLD, you have to first identify RLD, name of the RLD. If identify, after that, if go through the label of the RLD, in the label there is a description of API and composition, qualitative composition is there, not quantitative. Then storage condition, packaging material, then there is a number of NDC code to identify the product in US. Then for Europe market, there is a marketing authorization number of the product. For Canada market, drug identification number of the product. Different number is there for as per their regulatory market. We have to identify. Then when you go for the with label, then thoroughly you have to, if there is a patent is there through the Orange Book, patent not expire, then you have to do that, whether the patent, when patent will go for the expire. After that, you have to read all the patents. In patents, you can some identify some process, but not that much useful during the development for the product. And if you go for the summary basis of approval, the FDA side, there's different segment is there. Chemistry review. In chemistry review, the structure of the API, active pharmaceutical ingredients, what are the composition, specification is there. What are the specification that RLD already mentioned, RLD already submitted, then you have to follow that. You have to maintain all the limits, all the specification, all the parameters. Then after that, if the product is too much that tricky, the suspension, that emulsion, then you can go for that research article, some stability, some process, some dissolution parameter, how we can identify like that. Then when we have completed our literature search, then formulation strategy you have to prepare. After formulation strategy, you have to prepare the regulatory strategy, how we can file, and before development, IPS strategy also we have to prepare, whether the patent is there or not, patent, when patent can be expire, each and everything we have to analyze before development of any product. And this is the QVD. For ophthalmic products also is requirement is the QVD. QVD, that quality target product profile. Everybody know that whatever the RLD parameters, that a product will be the sterile, product is the ophthalmic dosage form, some that chemical parameters of the product, product will be the sterility of the product always sterile, then bacterial endotoxin. All these parameters is the quality target product profile. And after that, quality target product profile you have to complete. then identification of CQA, critical quality attributes. What is in your product? Then each and every critical quality parameters, you have to prepare the risk assessment. FDA always asks each and every parameter, you have to submit the risk assessment to produce a robust formulation or robust product in industry. Then after, when you prepare the risk assessment, all this thing, then go for the risk mitigation. How, if there is a risk, there is high risk, medium risk, low risk. If low risk, there is no need to go for the justification. If there is high and moderate risk, you have to give the proper justification. You have to give the proper data, how you can that parameter you set, and what is the precaution you have taken, what are the upper parameter and lower values of this product. Then when all this thing is established, then you prepare the control strategy for your product and process. You have to submit that control strategy to your production team. That if you go for the pressure for suspension, any suspension product, that particle size, then you have to mention what will be the high pressure, what will be the low pressure, and what will be the optimum pressure. Because in our lab, we always prepare some 500 ml, 1 liter, 2 liter batch. When you work for the industry always thinking how we can prepare from 5 liter to 100 liter, 200 liter. Always commercial thinking of industry. Industry always having the commercial thinking. How we can market the product, how it will be beneficial for the company. If you will go for the 5 liter, 10 liter, this is not the beneficial, because you have to take the 100 number of batches, 200 number of batches. This is not efficient for a company to run 24 hours that for single product. So we have to make a 100 liter, 200 liter. For 100 liter, 200 liter, so that parameters are very important. That what is the upper value, what is the lower value, that capture whatever batch size we have to propose in for production and for FDA during commercialization. That upper value and lower value, that parameter has to be play a key role during our development and submission of the product. When you go for 200 liter batch size, because if you prepare the exhibit batch size 50 liter or 100 liter, if you prepare the 200 liter batch size, if you have used the pressure, high pressure, 10,000 psi or 15,000 psi in exhibit batches, for 200 liters, if you just go for 20,000 psi for 200 liter batch size, you can't do. You have to do a control. What are the risk assessment is there? How, if we prepare 10, 15, 20 batches, how we can give the robust process and robust formulation with this parameter, how we can give that same product as well as R&D. Then we submit, we have to take the 3 batches, all the data with consent of regulatory, we have to submit the FDA. Then FDA review all the data with respect to our exhibit batches, then they can approve our product. For formulation development, if ophthalmic product, first we will go for the feasibility trials. Feasibility trials means just like if 100 ml, 200 ml, 300 ml batches, just whether the drug soluble easily or not, whether the product something that pH is little bit hydroxide, little bit HCl is required, or more HCl is required, whether 0.1 normal, instead of 0.1 normal, 0.01 normal, instead of 0.01 normal, if 1 normal can useful. Like that feasibility, we can take some 2 to 3 trials. When feasibility trials okay, then we go for the prototype formulation. Prototype formulation, already we have a formulation approved from FDA. There is a plus minus 5% composition with respect to the RLD. Then prototype formulation, after prototype formulation, we have to prepare 500 ml batch, 1 liter batch according to our analytical department requirement. For method development, how much quantity they required. So according to, we have to prepare the batches. When method development completed, then we go for stability study of product as per our proposed closure container system. That 3Ps, that LDP bottle, HDP bottle, PP bottle, LDP nozzle, or HDP cap, like that. Then that cap of this product, if you go with FDA site for different disease, the color of cap is already mentioned in regulatory site. If you go with the exhibit batches in white color, pink color, like that, nothing to worry about that. But when you go for the commercialization for different disease, different color of cap is there. For steroid, you have to mention that pink color. For that antihistamine, you go for that red color cap, already mentioned in regulatory different regulatory sites. Then when stability for lab batch, we have go for the initial 3-6 for accelerated, or long term 3-6-9-12. We are not doing any intermediate condition for lab batch study for long term as well as accelerated condition. After stability study, if 3 months stability data is okay, with respect to stability data, we just thorough review our data, and whatever the dose, maximum daily dose of your product, we have to establish our impurity limit of our product. If your maximum daily dose is 10 mg, then I think as per your ICH, you have to some 0.5, 0.2, that known impurity and unknown impurity, like that. If beyond ICH limit your impurity is showing, then you have to go with RLD data. RLD data means you have to analyze your initial RLD data as well as near expiry RLD data. If near expiry RLD data also shown beyond the ICH limit, then you can justify your impurity limit, although it beyond ICH. So RLD data also most important, that RLD initial as well as RLD near expiry sample, as well as ICH impurity limit also important. According to limit, that assay, pH, that first we will go with the different parameters. Then I'll give the specification. What we are writing in our specification. Then when specification decides, then we signed our specification. They go for that analytical method validation study. Whatever process method already developed in our analytical department, all the method we have to validate. After completion of validation, we have to transfer that method to our production plant. After production, when product that method already transferred, completion, then we'll go for process optimization in plant, because we have already taken in our lab maximum 5 liter batch size. But in production plant, 30 liter tank is there, 50 liter tank is there, 100 liter tank is there. Then for process optimization, we can decide whether if there is a solution, then easily we can go for the 30 liter, but exhibit at the time of exhibit is 50 liter, 100 liter is okay. If there is a suspension in emulsion, process optimization batch size will be the same as exhibit batch size, and that commercial batch size also same as exhibit batch size. Each and every company, maximum company follow this thing, because this is a complex process for the suspension and emulsion. So whatever batch size we have decided in exhibit and process optimization, commercial also same thing. If so, that's why previously, according to our discussion with the business development, what is the commercial requirement of the product in US and Europe? Accordingly, we decide our batch size of commercial and followed by that exhibit batch size for suspension and emulsion. For solution, because this is the easy process, you can take the 50 liter of batch size, you can take the 100 liter of batch size, because this is the mixing parameter you have to maintain, so nothing to worry. So this is very easy for solution. If your pack size is 2.5 ml or less, minimum batch size you have to you have to take 15 to 30 liters. If more than 2.5 ml pack size, minimum batch size will be at plant 50 liter, minimum 50 liter. This is according to our FDA guideline. Then when process optimization is okay, as well as parallelly whatever components we are using at plant, we have to do the compatibility study at our lab, that whatever tubings we are suggesting during the filling, during the transfer, what are the SS vessel we are using, 316L, what are the filters we are using, what are the gasket we are using at our plant. We have to do the compatibility study at different time points. Suppose initial 12 hours, 48 hours, 72 hours. Then we have to establish the data that our product is compatible. Whatever we have proposed the components up to 24 hours or 48 hours, we have to suggest our production. The product can be stable if you filling with that tube 24 hours, there is no absorption, there is no discoloration, all these things. So the compatibility study is most important. Then bulk holding study. Bulk holding study we are doing at lab as well as plant, because bulk holding is most important. If you prepare the batch as a 100 liter batch size and go for the filling, something happened in filling machine during filling, that rectify the filling machine sometimes needed, that some 10 hours, 5 hours, 6 hours, whether our product is stable up to that 12 hours, for 24 hours or 48 hours. So for that establishment, we have to do the bulk hold study at our lab as well as in our exhibit batches. Then go for the photostability studies. Photostability studies we are doing that what are the primary packaging of the product. Primary packaging means without label, without carton, without leaflet, just the 3-piece vial. That is the primary packaging. Then marketed product, then controlled product. Different lux hours, and we have to do the study whether our product is stable in photolytic condition or not. If the product is stable, there is no mention. If the product is not stable, then you have to write in your label, protected from light. Then thermal cycling study we are doing. Thermal cycling is most important. The product during transportation. If your product, thermal cycling we are doing like that product go for the -20 for 3 days, then thawing at our room temperatures. And this is one study. Then -20 degree for 3 days, then 40 degree, 25% RH, another 3 days. Like 3 cycle, 4 cycle we are doing. And we do the analysis with respect to related substance, with respect to assay, with respect to description, with respect to other physicochemical parameter, whether our product is okay or not. If product is okay, then the transportation is more easier. Otherwise, you have to mention during transportation, you have to store that product that 2 to 8 temperature or 2 to 25 degrees centigrade temperature, like that. Then filter sizing and filter validation. Then in our lab, we are using that disc filter, that 47 mm. 47 mm, but how you decide what is the size required during the filtration of 50 liter or 100 liter of the drug product? So for that, we have to do the Vmax study. Vmax study is filter sizing study. At different time interval, what is the throughput you are filter with particular your drug product. Then there is a one software is there. This will done by that our Millipore, Paul Corporation, MDI, Sartorius, and they suggest us you can go for that 5-inch filter for 50-liter batch size, 10-inch filter for 100-liter batch size. According to we write down in our master formula record and submit to our production. And followed by that, when filter sizing is over, before execution of exhibit batches, we have to do the filter validation study. Filter validation means because this is our ophthalmic products, it cannot go terminal sterilization. You have to sterile your product through filtration, because the melting temperature of your primary packaging material is near about 90 degree or 110, but for terminal sterilization you have to do 121. So the packaging component will be deforming. So that's why always for ophthalmic product, always we prefer the filtration, sterilization by filtration. For sterilization or filtration, we go for the filter validation study. One is from bubble point study. Another one is compatibility study. Another one is bacterial challenge study, and another one is extractable study. That all the study are doing at Millipore or Paul or Sartorius, different vendor, whatever you have decided. Then they have submitted their report, whether product is compatible, all these things. Then go for further procurement, and the documents also required during the filing to our regulatory agency. Then we'll go for the preservative efficacy study. Preservative efficacy study, because we are using in ophthalmic benzalkonium chloride and that benzododecinium bromide, then SOCl2 like that. But when you go for stability, some quaternary compound on stability, their assay will be the less, near about 85%, 86%, or 90%. So to give the specification limit, we have to do our preservative efficacy study. How we can do the preservative efficacy? That we have to make batches with different concentration of preservative, 25%, 35%, 75%. If our product with different microorganisms will challenge our 50% or 25% concentration preservative, if that pass with different microbial challenge, then we can decide our specification. If our product pass to 50%, then we can decide that if in stability our preservative is less than 75%, it can work. That preservative efficacy, it will act as our to set our specification limit during submission and during stability and shelf life. And another one thing is in-use stability study. I have not written here. In-use stability study you have to do, because if you see any PIL or carton of ophthalmic products, they have written after 28 days you have to discard the vial after opening. So during 28 days, we have to go for the in-use study. How we can do the in-use study? First day that we have to open our cap, and whatever doses written in PIL, that label, that 2 drops in evening and morning, then we have to discard the 2 drops and keep it as such. Like that, we have to do up to 28 days. After 28 days, we go for the analysis, that assay, RS, preservative efficacy, then endotoxin, all these things. If the parameter is okay, then we can suggest that our product is safe up to 28 days after opening of the cap. Otherwise, different time interval you can do, that 15 days, 28 days, 32 days, 45 days. If your product is suitable for 45 days, you can claim in your label that our product after opening our cap, you can use up to 45 days. After that, you can discard. This is the drug product quality test for any ophthalmic products. For appearance, nothing is, this is a description of the product. Viscosity, surface tension. Viscosity, for if there is a viscosity agent is there, xanthan gum, hydroxyethyl cellulose, carbomer, then you have to do the viscosity and viscosity at different shear rate. Whether your viscosity will change or not, you have to prove. Then osmolarity, pH, whatever container you suggest to during commercialization, that the drop size of this container is matching with RLD or not, with respect to 45-degree angle and 90-degree angle. 20 drops, 10 drops, first 10 drops, then 2 drops 10 times, 3 drops 10 times. Each and every drops you have to measure by weight and compare with the RLD product. Then assay of API and preservative. Then impurity profile of active. Then particle size distribution and globule size distribution. Particle size distribution, this is most important in globule size distribution, also most important for emulsion and suspension, because for population bioequivalence, the span value and D50 is the most important, and for nano emulsion the range is too short. So the accuracy of this globule size and particle size most important. Then particle morphology, that J3 morphology. Then dissolution. For dissolution, we are using the apparatus of type 4 or bottle rotating apparatus. Then sterility test, container closure integrity. And container closure integrity we are doing in stability, because if you go for sterility test, it will take 14 days. Then when you submit the product after 6 months of accelerated study, then again you have to wait for the 14 days. So to skip of the study, we are proposing container closure integrity. If your container closure integrity is okay, the product will be sterile. So we suggest that you can do your product sterility on 12 months, 18 months, or 24 months, and 3 months also. But during submission, we are doing the container closure integrity and go for the submission for FDA or FDA accept. Then extractable and leachable. This is the most important thing, because we are using the semi-permeable container. With semi-permeable container, you are using that label, you are using the glue, you are using that carton, leaflet. That color goes inside and outside. It can harmful for your patient. So as per regulatory requirement, you have to choose the adhesive, you have to choose the color, the 7 Pantone color, all these things. The packaging development we have, they are deciding, and all this, everything, that label, that leaflet, glue, container, and then ink, we have to submit a different lab to extract the components and different worst case condition. When extractable, they have identified ability for ophthalmic product at definite time interval, that 6 months, 18 months, or 24 months. We have to send the sample that particular contact testing laboratory with respect to the extractable, whatever they have identified in our product stability, that some extractable that leach out in our product during stability or not, we have to identify. Then irritation study, toxicity study, preservative efficacy, already I have mentioned. Water loss study. Water loss study, because if you go for the injection is the glass wire, there is no chance for water permeation. But when you go for the semi-permeable container, there is a chance of water permeation or water loss. If you use the 5 ml pack, after 6 month accelerated condition, it will comes to 4.8. But as per USP, your pack size should contain not less than 5 ml. And how you decide? So for water loss, because this is the 5% water loss as per ICH guidelines for semi-permeable container, not more than 5%. When you go for the filling, we already added the 5%. That another 5% means 5.25 ml. This is our lowest fill volume. Then we can go for our machine tolerance plus 2% and plus 2%, that upper volume. Then during the water loss instability, your product not less than 5 ml, it will come definitely 5 ml. Like that, we have to decide our fill volume in our container. Then for complex ophthalmic products, you have to do the population bioequivalence. If you go the FDA side, each and every product they have given a bio guidance. Whether you go for in vitro characterization, whether you go for the in vivo characterization. For ophthalmic products, maximum ophthalmic products, FDA suggested you go for the in vitro characterization. Because of in vivo characterization is too tough. Go for that. So each and every where you have to go, some study in aqueous humor, some studies in dry eye. So they are suggesting you go for the in vitro characterization. If your in vitro characterization parameter is okay, then you can proceed for FDA submission and commercialization. So in vitro characterization, with whom compare? You have to compare with the innovator, that RLD product. Whatever description, pH, that viscosity at different shear rate, buffer capacity, specific gravity, then globule size or particle size, then 10, 20, then scan value, PDI, Z average, everything is important. Then one thing is important is opening torque and closing torque. Because when you go for the stability study of the any product for ophthalmic, you have to do stability study both the orientation, upright of the product as well as inverted condition. If your product container, the torque is not optimum or torque is not good, that there is a chance of leakage of the product when you will go for the inverted condition. If there is a leakage, then your water loss study will be failed. If there is a suspension, there is a leakage, some particles some solution will be comes out and dry in caps and nozzle, then it will harm to your particle size or delivery of the product. So opening torque and closing torque is the most important parameter during filling. Then dose accuracy, 25 drops and 25 drops in cool condition, 2 to 8. That standard condition, what is the product temperature? Some warm condition we have to do. So the stability study we have to perform for all the products, and stability temperature, whatever mentioned in ICH, you have to follow the guideline, that Q1A stability testing of new drug substance and product, then stability testing for stability of new drug substance and product. Then if your product is 5 ml, 2.5 ml, 10 ml, you can approach the bracketing approach during the submission. I can do the stability throughout all the stations of 2.5 ml and 10 ml, that minimum pack size and maximum pack size. Then whatever the optimum pack size, the 5 ml, you can do that only accelerated to station or long term. It will be the called as the bracketing study. Then evaluation stability data packaging for registration applications, the different climatic condition. All these guideline in as per ICH we have to follow. This is the long-term different temperature condition as per the stability test method. The long-term, that above table, this is the general, and below table, this is the for semi-permeable container. If you see, the difference is the RH. If you accelerate condition for semi-permeable, 40 degrees centigrade, not more than 25% RH. But in above table, the accelerated condition 40 degrees centigrade, 75% RH. Why this 25 RH in for semi-permeable because the permeation is there. So this is the worst condition for semi-permeable container. You have to perform stability study in low RH condition. Then you can get the actual water loss. If you do the you do the stability study in with above condition, then during water loss study, whatever results will come, you have to multiply with some factors 1.6 or 1.7 like that, then you can give the results. But if you maintain that 40 degrees centigrade not more than 25%, whatever water loss come, you can give the same data, it will be okay. But for 75% RH, some factor is there, you have to multiply with the result, then you can submit the data. For formulation development specification, first we have to prepare the API specification. API specification, if the product is as per compendial USP Europe, then we have to maintain all these things. And before doing any development, whether your drug substance qualify as per the impurity, as per ICH, as per compendial, then you can go ahead for that. Because otherwise it will be very hectic to maintain the impurity limit during the stability. Then excipient specification as per compendial monograph or as per ICH requirement. After everything is okay, then as per our stability data, as per our RLD review data, all these things we have to prepare in process specification. In process means when the batch is prepared before filtration, the bulk solution, whether the assay is okay, whether the preservative content is okay, whether the pH is okay, this is the in-process specification. At the time we are doing the bioburden, what is the loaded is there, bioburden loading is there, because there is a non-sterile, there is unfiltered solution. After that, when that product is filtered and sterile, then product is go for the filling. That sample is called the release specification. Release specification, you have to maintain all the parameters for your product. Apart from water loss, all these things. And for shelf life specification, that you have to include the water loss study, and as well as for release specification, you have to mention the residual solvents data and elemental impurity in release specification, whether your product is passed through that all this guideline or not. Then go for the shelf life specification. How to simulate the product? Characterize the similarity of the product. One is qualitative similarity, whether the product is qualitative okay or not, whether your product is qualitative or not. Then quantitative, I have already mentioned that we have to submit that FDA to our formulation before finalization of formula or before doing the development with respect to our RLD, whether our formulation in comparison with RLD plus minus 5% is not, or yes or not. If plus minus 5%, FDA will go ahead. If plus minus 5%, FDA will reject, then you will again work, again submit, wait for another 3 months for response from FDA, then again development start. Then structural similarity, same components, same concentration, same arrangements of matter, microstructure, all these things. These three characterized similarity you have to maintain. Then formulation document, this is the pharmaceutical equivalent. The qualitative, quantitative, the same ingredient, same dosage form, strength, quality, purity, identities, everything is same. Packaging configuration also same. Packaging component MOC, material of construction of packaging component, it will be the same. Then expiration time, if the your RLD 24 months, then your product will be the 24 months you have to shelf life. And whether product is bioequivalent, for bioequivalent you have to, for ophthalmic, maximum ophthalmic complex. For solution, there is nothing required. You have to compare the 3 lot of RLD and 3 exhibit batch. 9 times you have to do the physicochemical characterization and submit the data is okay. For suspension and emulsion, you have to do the in vitro characterization for bioequivalent or not. The packaging design, already I have told that 3-piece and as well as your unit dose. Nowadays, one container is there, preservative-free container. Preservative-free container, this is the multi-dose container. There is a tip, there is a mechanism, patented mechanism. The company name is Novelia. They are providing. The product is whatever product through the container will be delivered will be the sterile. They claim and they proved. According to the viscosity of the product, the nozzle will be decided by the company. You have to send a sample to the packaging development company. They will suggest which nozzle you can use. According to you have to procure for that product, then that preservative-free bottle you can use. This is the new trends all are using because this is very useful and the shelf life will be the more and easily the same machine, whatever machine we are using for 3-piece filling, that can be used for that preservative free bottle. So that's why this is more useful nowadays. All are using all these things. This is the unit dose, that refresh plus all these things, cyclosporin also, Ristasis, that unit dose is there. But if you go through the FDA site, previously 5 to 8 years before that, they are using that unit dose. Now they are also using that preservative free container, that multi-dose 5 ml container. They are using both both the pack size are available in FDA site. You can go through the FDA site, then it will be easily you can see. The importance of cross-functional department during our product development and life cycle management during our product. That's for business development, analytical development, packaging development, sourcing, manufacturing, quality control, quality assurance, regulatory affairs, and marketing. And middle, we are the formulation, research, and development. This is the base, and others, all others are the supporting department. Business development, they are providing or they are identify the drug product. They give the opportunity to do the development. We go for the development, analytical development or supporting department to analyze our product, packaging development or supporting department to analyze our packaging material and what are the packaging material we can use our development. Then sourcing is the most important. Manufacturing, quality control, quality assurance, regulatory affairs. Regulatory affairs main during the filing, during the that correspondence with FDA. Any correspondence with FDA, regulatory affairs department is the most important department, and for submission also they are the most important. One case study in my career, that so many projects I have done. If you till some prednisolone suspension, Tobradex suspension, Ciprodex suspension, cyclosporin emulsion, that difluprednol emulsion, loteprednol suspension, loteprednol gels. I have so many products I have done for different companies. This is one case study. This is a tobramycin and dexamethasone ophthalmic suspension. That brand name is Tobradex. Dosage form of ophthalmic suspension, strength is 0.3, 0.1. 0.3 for tobramycin, 0.1 is dexamethasone. The pack size is 2.5, 5 and 10 ml. This is a steroid responsive inflammatory ocular and condition for which. Now you can ask me, I have told that for steroid the pink color cap you have to use, but innovator here is the white color because the product is approved that 10 years, 12 years, 15 years before. That regulatory daily change their norms and they are updating. So now whatever steroids you have to prepare in, you have to mention that your cap will be the pink color. In challenges for this product is product challenge and process challenge. In product challenge, the composition is 0.3 and 0.1. and one drug is suspended form and one drug is solution form. There is a chance in segregation because the particle size is less than 5 micron and D10 is less than 1.6 micron. Product is not that, because this is suspension, the product viscosity is 4.5, and the viscosity agent is used hydroxyethyl cellulose. If product viscosity 4.5, the sedimentation will be the more within 5 days, 10 days sedimentation is there. So it is also challenged to redisperse and uniformity content. Then often each drop should be delivered desired dosage. Each and every drop you have to analyze that 2 drops you have to take and go for the assay, and assay will be the same. So this is also one challenge. And for process channel, particle size distribution, multi-tier specification. Multi-tier specification means you have to maintain D10, D50, D90. If D10 more, D90 less, then it will impact on your span value, and it will impact on your population of bioequivalence. Then it will not pass in vitro characterization, and you cannot submit your product in FDA. Then particle size reduction to be achieved in situ during the processing. That product slurry is non-sterile. We have to sterile. For sterile, how we can sterile the slurry? After sterile, how can we do that particle size reduction? This is the most important thing. Then process should be able to produce the sterile product, because this is a 3-phase. One is viscosity agent, one is tobramycin phase, one is your dexamethasone phase. You have to 3 phase. You have to maintain the sterility. You have to mix. You have to do the particle size reduction. So product sterility also important. And whatever machine we are using and whatever tanks we are using, then this process itself 24 to 36 hours for manufacturing of bulk to filling. So there is a chance in elemental impurities. So how we can minimize of that? This is also important. And the process should not result in high temperature that will degrade the drugs. If your dexamethasone, you have to sterile 121, and your polymer also you have to dissolve in high temperature, and you have to down the viscosity with the help of the polymer that are in situ. So how you can manage the temperature that will not impact on your product quality or impurity of the product. Then, after all, development of this product process was challenged. We developed a novel, innovative, and reproducible, cost-effective process. This is our achievement. And this for particular product, this is the interesting thing. When we are developing in our previous company, there is no guidelines for this product. Then we have to do this thoroughly study that describe each and every parameter, particle size, Z3 morphology, everything, viscosity at differential rate, drop size angles, all these things studied. Then we prepared a small book-like thing, and we submit to our FDA for pre-ANDA meeting, because this is a 2-drug product. If you go for a bio study, it will very big challenge as well as very cost. If some product is in vitro characterization, whether we can go for the in vitro characterization or not, then FDA asks what study you have done. Then we submit all the data with comparative study with RLD, like 100, 120 pages we have submit. After reviewing FDA call a meeting with our top leaders. Then they assure these are the parameters we are maintaining. The product quality will be the same as per RLD. Then FDA published Topradex in vitro guideline in FDA site. Whatever in vitro guideline in FDA site nowadays, we saw that guidance preparing by us at the time of development, and we have submitted to the FDA. After our submission, that in vitro characterization, that guidance comes to in FDA sites for Tobradex particularly. How we overcome the challenges? I have already told, modern analytical tools like laser diffraction. We are using that Malvern as well as Beckman. Both the machines, they can capable to do the analysis of particle size. We are doing in both the system, whether if the system will change or particle size is any impact or not, we have to check. Then G3 morphology, then HPLC, particle size analyzer, and viscometer. Viscometer, we are using the general viscosity for Brookfield, or for viscosity, the rheological behavior, we are using the Anton par. Then here is the source is the most important. Our sourcing team has done a great job. Right source identify and provide desired quality of the requirement as per FDA and ICH and USP requirements. And also product is ophthalmic eye drop. The process should be ensured that the sterile, because 36 to 40 hours process, so many phases we are mixing, 3 to 4 phases, we have to maintain the sterility of the product. This is a big challenge. So we overcome. Now I think that product will be commercialized. Some queries that we had received in my previous company, they are sorting out. I think it will. Then some particle size distribution, critical quality attributes, the D10, D50, D90, not for only one analysis. You have to do 20 times for 3 lots, 3 exhibit batches. 20 times you have to analyze and your result will come same. Then your result will pass population bioequivalence. That's span value as well as D50 value. This is the most important. So your product will be the reproducible, your particle size will be the uniform throughout the process, is the most important. Then achieving right size macroparticle, particle size, and particle size distribution in the final product was a challenge. I have already told. And process equipment selected was required to ensure consistent shear levels and repeatable results. Also, there should not be significant increase in temperature, and that no elemental metal contamination occurs during the processing due to high shear and high pressure. Because so many things we are using, milling technology we are using, and high pressure, that different high pressure, that microfluidics, that all this machine we are using. But which machine is suitable, then we decide according to our developmental data, then we'll go ahead and to minimize that metal contamination. Then we selected the process product stream through precisely engineered micro channels within the unique interaction chamber that has the ability to have control shear rates. Then this repeatable process results in tiny particles with a uniform distribution, and scale-up performance from lab to product is granted. We took the 5 liter, then 30 liter, then now 100 liter batch size. And before taking exhibit batches for submission batches, we took near about 40 to 50 scale-up batches, whether our product process is reproducible or not. So this is the most important. When you go for the submission batches, there is no OS, OOT, there is no process difference. So we have made so many scale-up batches to boost our confidence, and we can make the batches in during execution, and it will be very good. It will give the very good results, and we got the results very good. Then final product was able to uniformly deliver both the low dose drugs in each drop, reproducible process, produce sterile product, then process short manufacturing time, lower cost of manufacturing time, because previously it was we'll go for the different machine and different thing, then 48 hours, 52 hours, like that. Our 3-4 scientists that they are from morning to evening to night, they are doing what a big job they have done. So how we can reduce the process, how we can reduce the particle size with the pressure, more pressure or temperature, like that. So many combination we are using, and we got the results, very useful results. That's all. Any questions?
Thank you so much, sir, for providing such informative knowledge about ophthalmic products, its fundamental aspects for development of formulation, with regulatory consideration that what type of way we are supposed to consider about the Orange Book listed, reference listed formulation before starting the development of generic
Yes.
formulation in terms of ophthalmic product. Thank you so much
Yes.
on behalf of Faculty
Yes.
of
Yes.
Pharmacy, Parul Institute of Pharmacy, Parul University for delivering the session.
Thank you, ma'am. And I request you, ma'am, and one
Yes,
thing
yes,
for M.Pharm students and
yes.
for PhD students, if they are capable to do search all the regulatory and FDA side, because for company, because all company we are taking so many interviews for fresher candidate because they
Yes,
are what is the tablet, what is the capsule, but nowadays you have to get the knowledge some regulatory. What is the ICH guideline? What is the stability parameters? Or what little
it is
things?
required.
It will be more
It is
useful
really required.
nowadays.
Huh? Yes,
It is required. It is really
yes, yes, yes.
required. If you go through the
Before
FDA
starting
sites,
the
it will be very
project,
easy.
we should first thoroughly understand reference listed formulation about what they are going to develop the product.
Yes, 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 yes.
Yes, yes,
Definitely,
yes.
definitely. If you want, then I can, anytime I can help you. How you can go
Yes, yes.
through, what are the channels to follow. It will be beneficial for your students before interview for any companies.
Yes, yes, sir. Definitely, sir. Absolutely.
Thank you.
Thank
Thank you.
you. Yes.
Thank you. Any question? That's all, ma'am.
Yes, yes, yes. No issue, sir. If something will have, we will
Okay.
definitely
Okay.
draft in your mail and you can reply also.
Yeah, definitely, ma'am. I will help you.
Thank you, sir.
Okay. Thank you. Thank you.